Okay, so I had a little technical difficulty with the last lecture recording, so I stopped here. Now, I was going to talk about the pigments and lab absorption, but I believe we talk about this in the lab, so just to avoid the repeat, I'm going to skip the next few slides. And I'm gonna mention a little bit about the absorption spectrum, just in case you didn't get a good understanding in the lab. So there are different pigments in the leaves that can absorb sunlight and then capture the energy in the sunlight. So there are a couple types of main pigments. So we have chlorophyll and then we have keratin. So chlorophyll is the main photosynthetic pigment. So you can see in this graph that chlorophyll has the highest absorption in the blue light range right here, and then in the red light range. So that means that these pigments utilize these lights the most for photosynthesis. Okay, so they like these two ranges the most. Okay, smiley face. And then you can see that the absorbance for the green light range is very low, right? This is the valley of those lines. So that means those pigments do not like green light, okay, around this range of wavelengths right here. So what the pigments do is that they absorb what they can use for photosynthesis, meaning these two, and then they reflect green light back because they don't use them. And then when the green light is reflected back from the leaves and enters your eyes, that's how you perceive the leaves as green or algae as green. Okay, so the pigments really use colors other than green for photosynthesis. Okay, so this is about the absorption of light for different photosynthetic pigments. Okay, I'm gonna mention this real quick because it has real-life applications. So we talk about chlorophyll, and they reflect the green lights back, so that's why they appear green. We also have carotenoids, so that's another group of pigments that you often found in plants. Now, these carotenoids have a range of colors, so they can be yellow or orange or red. So they are often found in certain fruits and other plants. So the red color in tomato, yellow from corn seeds, or orange from orange peel. So all those colors come from different carotenoids. All right. Now, usually leaves have chlorophyll and carotenoids. Okay, these colors, but chlorophyll is the most abundant pigment, so that's why those leaves will be green. Okay, because chlorophyll kind of masks the other pigments, so their colors just don't show. But when we get to the fall, when the temperature drops, the leaves are going to lose the chlorophyll first, right? Remember, the trees, they're going to shed their leaves to kind of reduce the chemical reactions, reduce the metabolism so that they can survive through the winter. So when they lose those leaves, or about to lose those leaves, the pigments in the leaves, the tissues, the cells will start to break down. So chlorophyll will be broken down first. Once chlorophyll is gone, now the other pigments start to show. So you're gonna see other colors from pigments like carotenoids, right? Those other pigments. So this is what causes the leaf or fall leaf color change, right? So just some interesting facts. Okay, so how does photosynthesis convert light energy into chemical energy? So you have to remember this. Those plants, algae or cyanobacteria, they are converting one form of energy to another form of energy which they can actually use. So they're going to convert light energy into chemical energy. So chemical energy just refers to the energy that stores in chemical bond. Okay, so they're going to be temporarily stored in chemical bonds, and then when you break the chemical bonds, you release the energy for other cellular activities. So there are two steps involved in photosynthesis. The first one is called the light reactions. Now, some people call it light-dependent reactions. That's fine too, because this step does require sunlight. So that's why we call it light-dependent reactions. So the purpose, the goal for step one is to harvest solar energy. So you're going to have those pigments and some other proteins to extract the energy from sunlight, okay? And then you are going to convert that energy into a form of chemical energy, okay? And that's going to be two things. ATP, you're familiar with ATP. The second one you might not know, so it's called NADPH. So this is an electron carrier, so they can carry electrons, they can carry energy. All right. So again, for the first step, you harvest solar energy, and you're gonna make two kind of intermediate electron carriers, energy carriers, and they're ATP and NADPH. All right. Now these two things are gonna be needed in the next step. So the next step is called light-independent reactions, which means this process does not require light. So it can happen during daytime, or it can happen at night when there's no light because it does not require sunlight. So other name, some people also call it Calvin cycle. And then in this cycle, you are going to use ATP and NADPH produced from step 1 to make carbohydrates, glucose. Okay, and in this process, you also need CO2, carbon dioxide. Okay, so you're gonna need energy and electrons, and then you're gonna need carbon dioxide. So provide energy to synthesize glucose, and then to provide electrons so that the carbon is reduced to the carbon in glucose. Remember the reaction that we looked at earlier? Carbon is reduced, right? It's reduced over here. So you need to provide electrons to reduce the carbon, because when something is reduced, that means they're gaining electrons. All right, now that's the two general steps. So this is more just a repeat of what I just talked about, but the figure, there's a nice figure over here, and then there's some more detail. All right, so let's look at this detailed information here. All right, now this picture tells you a few pieces of information. So the step 1 light reactions take place in the thylakoid membrane. Okay, so what is that thylakoid membrane? Now this whole thing right here, this is a one chloroplast. Remember that cell, that's the cell organelle that photosynthesizes. So this is a chloroplast. Alright, now inside these chloroplasts, there are these stacked disc structures, this green thing. That's called the thylakoids. Alright, now the thylakoids are membrane-bound structures. So this is going to be the membrane. Alright, so the light reactions take place in the membrane of the thylakoids. Okay, now in the chloroplast, besides the thylakoids, you also have the rest of the space. Okay, so you have a fluid filling the rest of the space. So this space is called the stroma. Alright, so basically chloroplast. In chloroplast, you have inside, you have thylakoids, and then you have stroma. Okay, so the first step happens in thylakoids. So again, you have to make ATP and NADPH, and then in that process, water is split. And then when water is split, oxygen is produced. Okay, remember we talked about over here? Again, delete that and change that to 6. So oxygen over here in water becomes oxygen gas. So it loses hydrogen. Okay, so it's oxidized, right? So it loses electrons. So that's what happens here. Water is split, and then oxygen is produced as a byproduct. Okay? And then the second step we talk about, it's not dependent on light, so it can happen anytime. And the second step takes place in the stroma, so it takes place outside the thylakoids, okay, in the rest of the space in the chloroplast. And make sure you don't mix stroma with the stoma. So stoma is the openings on the underside of the leaf that can take in carbon dioxide, and the stroma is this space inside the chloroplast. Alright, the light-dependent reactions use ATP and NADPH and the carbon dioxide to make carbohydrates. So that's more like a repeat from the last slide. Okay, so we're going to talk about the details of how the chloroplast, the cell organelle, can harvest the solar energy and make glucose. We have to talk about the light reactions machinery first. So the machinery is called Photosystems, and sometimes we just really, never mind. We don't have any good abbreviation for that. So just the whole name, photosystem. We have 2 photosystems, so we have 2 and we have 1. So it doesn't mean that the reaction goes from 1 first and then to 2. It doesn't really mean that. The 1 and 2 are based on the order of when they were discovered. Okay, so photosystem 1 was discovered first, so we named it 1, and then 2 was discovered later. Okay. So a photosystem consists of 2 things, light harvesting complex and reaction center. So you don't have to memorize that, okay? And in the light harvesting complex, that's where you find the pigments, okay? So basically, remember, in the photosystem, you have a pigment, and then you have a reaction center, all right? So the pigments can pass light energy to 2 special chlorophyll molecules. So there you go. So the 2 main pigments that are used in photosynthesis are chlorophyll A and chlorophyll B. Okay, so over here we focused on chlorophyll A. So chlorophyll A receives the sunlight, the solar energy, and then the electrons in the chlorophyll A. So these electrons, as you can see, this is the electrons right here. So when the light hits the chlorophyll A, the electrons of chlorophyll A get really excited because they receive all the energy from sunlight, right? When you have sunlight kind of shining on you, you can feel the heat, right? You can kind of feel the energy in the sunlight. So when the chlorophyll gets the energy, the electrons, as part of the atoms, they get really excited, and then they are going to move on to the primary electron receptor. Okay? So they will be passed on to other membrane enzymes. So they will be passed on. Okay? And eventually they will be used to generate the things that we need for step 2. But over here, so these electrons are passed on, right? So they left the chlorophyll molecules, but you need to replace those electrons so that you can have more electrons get excited and be passed on in the reaction pathway. So what we do is we're gonna split water to hydrogen and oxygen gas. And remember, in that process, oxygen is oxidized. That means oxygen loses electrons. So the electrons lost from oxygen will be coming here to replace the electrons that you lost from the excitation from sunlight. Okay. All right. Here is actually a better picture. All right, so let's go through the text real quick, and then I can point out the different components in the structure, or in this figure. Now, this can be very complex, so just bear with me. I know it's a little bit boring, but we have to go through this. Okay, so we talk about the pigments absorb energy from sunlight, and the photosynthesis is initiated. Okay, so don't worry about these terms and diatomic pigments. Don't worry about that. Just remember that when the pigments receive the energy excites the electrons. All right. Okay, so the electrons are going to pass through the electron transport chain, which will eventually pump hydrogen to move hydrogen across the thylakoid membrane. All right. Here, let's look at the picture. So here, let's see, over here, the electrons are excited, and then the electrons will be passed through this protein complex. All right, and then the protein complex will move the hydrogen against concentration gradient. Okay. So do you see the arrow right here? So hydrogen ions are moved from, if you remember, this is the stroma, and then this is the thylakoid, right? So inside the thylakoid, we call that space lumen. So this is thylakoid lumen. So based on the arrow, you can tell that the hydrogen ions are moved from stroma to the thylakoid lumen, right? And then you can also tell that you have a lot of hydrogen ions in the thylakoid lumen on this side of the thylakoid membrane, but you don't have too many hydrogens on this side. So the transport of hydrogen ion is actually against concentration gradient. Against concentration gradient. So that means you need energy, right, to move the hydrogen ions. So this is a type of active transport. So the energy comes from these excited electrons. So they will provide energy for this protein complex to move or to pump hydrogen ions from low concentration to high concentration, all right, to build a hydrogen concentration gradient. All right, now this action builds up a very high concentration of hydrogen ions, and then on the nearby area of the thylakoid membrane over here, you're going to have the secondary active transport. Secondary active transport. Now, I don't know if you guys remember, so secondary active transport does not require direct energy input, right? Because it actually uses the energy you invested in here, the primary active transport. Okay, so what happens is these hydrogen ions, you have high concentration on this side and low concentration on this side. And then there is a protein that allows the hydrogen ion to pass through. So now everything is good. The hydrogen ion is gonna diffuse, it's gonna move from high to low following the concentration gradient. So the hydrogen ion is going to flow through this protein, which is called ATP synthase. So you can probably take a guess. ASE, that means it's probably an enzyme, and it synthesizes ATP. Okay? So as the hydrogen ion flows through this protein, it kind of powers this enzyme. So it's more like a water pushing a motor kind of way to get it to work. So you have ions flowing through. This powers this motor, which is the ATP synthase. So the ATP synthase, this motor is going to synthesize ATP from ADP and an organic phosphate ion. Sorry, not an organic phosphate group, not an ion. Okay, so now basically you store the energy from sunlight to ATP, which is the battery, right? I talked about before. So you can use the energy stored in the battery for some other uses. All right. So this is pretty much the light reactions. Now I don't really need you to memorize everything. So on the test, I will give you a picture very similar to this. So it's gonna include all the information that you need to explain the process. So you have to know that this is chlorophyll, that's the pigment. And then when the sunlight strikes the pigment, it excites the electrons from the pigment. So these electrons carry the solar energy and they will be passed through a protein complex. And then the energy will be harvested by the protein complex to pump hydrogen ion against concentration from the stroma into the thylakoid lumen. All right. Now the purpose is so that you can establish a concentration gradient for hydrogen ion. Because on the other side of the thylakoid over here, you are going to use that concentration gradient to power an enzyme, a motor enzyme that will synthesize ATP. Okay, so this is how you can convert the light energy into chemical energy stored as ATP. All right, so this is part of the light-dependent reactions. There is another part that I don't have a picture. I just have a quick description. So this is a photosynthesis too. So I'm going to say PS2. So this is a good abbreviation. I think some people do use PS to kind of indicate this is photosystem. Okay, so photosystem 2 will produce ATP. How about photosynthesis 1? Alright, photosynthesis 1 will also absorb a photon from the light. So just remember, also harvest light energy. And then what happens is it's going to make the other electron carrier. So that's the NADPH. Okay, so ATP has energy, and the NADPH is more like an electron carrier. So it's going to temporarily carry the electrons, and then eventually they're going to pass these electrons to the carbon to reduce the carbon from inorganic carbon to the carbon in glucose, organic carbon. Okay. All right. So let's see. There's a quick video. You can watch that video and then do. Ooh, I don't have the handout for you. So let me get the handout. I will probably send you guys an electronic copy because I don't have any money left in my printing account because they're having some issues. So I'll try to remember to give you guys the handout. Okay, just post it on Blackboard. Okay, now we talk about the photosynthesis step 1, which is the light-dependent reactions, and then this is the second one, the light-independent reaction. So light-independent, also known as Calvin cycle. So where is this taking place? If you remember, it takes place in the stroma, right? Not inside the thylakoid, but outside the thylakoid, still inside the chloroplast. And where do ATP and NADPH come from? So this process, this cycle requires ATP and NADPH. So these two things are produced in the previous step, right? The light-dependent reactions. Okay, now again, this is how we make glucose from carbohydrates. Okay, so again, this is more like a cyclic reactions because you can see things start from here, right, and then it goes through the cycle, and then you can come back and start making more glucose. So again, there are a lot of intermediate products involved, so I don't need you guys to memorize those things. I don't even memorize all those chemicals. So I just need you to be able to use the information in this graph and then talk about the process if I ask you. Okay, so again, you need to know how to read the graph. So the process starts with carbon dioxide, right? Remember, you are putting in carbon dioxide as the reactant. Okay, so you are going to have an enzyme, and then you are going to have another chemical. Let's use this picture. This is a little bit better, and then I have questions so that we can fill out the questions. Okay, so here's the enzyme that I was talking about that will catalyze the first reaction. Okay, and then we're gonna have another chemical molecule. So I put all the full names over here. So this chemical is the ribulose biphosphate. So ribulose with OSE, it's a sugar, right? So it's actually a 5 carbon sugar. So if you count 1, 2, 3, 4, 5 carbon sugar. And bi means 2. So this 5 carbon sugar has 2 phosphate groups attached to it. Okay. So again, you don't have to memorize the name. I probably will just give you this abbreviation. Just know what it does. So basically, it will accept the carbon dioxide, and then when they bind, you are going to use an enzyme, rubiscal, to catalyze this chemical reaction. Okay, so to kind of make them linked. And then they're gonna form a 6 carbon molecule, but that 6 carbon molecule is not very stable, so it's gonna immediately break down into this 3 carbon chemical called a PGA. So PGA is the 3-phosphor glycerate. So this is a PGA. So you can see it has 1, 2, 3. There are 3 carbon. All right. Now this first step is called carbon fixation, which means you are converting carbon from the inorganic carbon in carbon dioxide to organic carbon. Right? We talk about the differences between inorganic and organic chemicals. So now you have carbon-carbon bond. So now it's an organic molecule with a carbon. Okay. Now, the next step, you're going to try to reduce PGA, right? So you are going to give the carbon, electrons, and the hydrogens. So in this process, it requires energy. So you're going to put in ATP to provide energy. That's for the first step. So you need energy. And then the second step, you need to reduce PGA. So in order to do that, you have to provide electrons. And the electrons are provided by NADPH. Okay? So after it donates the electrons and the hydrogen, it becomes NADP+, right? Because it loses electrons, so now it carries a negative or positive charge. Okay? So that's the second step, reduction of PGA. And this is going to continue to go. It will be converted to PGAL, or G3P, that's the full name. And then this PGAL eventually is going to kind of split. So you have one molecule coming down here, which will become glucose. And I'll explain why there's the half over there. And then the majority of it, 5 molecules of PGAL will go back into this cycle. And then it's going to be used to regenerate this ribulose biphosphate so that you can use that in the next cycle to fix more carbon. Okay, so that's the general process. Now let's talk about why here it's a half. So if you remember the reaction of photosynthesis, so you have 6 carbon dioxide and the water, 6 water, and you get 1 molecule of glucose. This is 6. And then oxygen, right? So you can see in order to make 1 glucose, you need 6 carbon dioxide molecules. And that's because there are 6 carbon atoms in glucose. So each carbon dioxide only has 1 carbon, right? So you need 6 carbon dioxide in total. So if you put in 3 molecules of carbon dioxide, you're going to get half molecule of glucose, right? So that means in order to make 1 molecule of glucose, you are going to have to double this amount. So you're going to put in 6 carbon dioxide. All right, so that's the math. So let's answer the questions. Calvin cycle requires CO2, that's the reactant, but you also need the molecule that provides energy, which is ATP, right? And then you also need the molecule that acts as an electron carrier, which is over here, so NADPH. So these things, not carbon dioxide, but ATP and NADPH, are produced in the previous step called the light. Is it dependent or independent? Independent, right? The first step relies on light because you're capturing the solar energy. Okay, number 2, in carbon fixation, which enzyme catalyzes this reaction? So this is carbon fixation, and you can see this is the enzyme that will catalyze the first step, which is to combine the carbon dioxide and then the ribulose biphosphate. Okay, so the enzyme is, Okay, give me 1 second to plug in my computer. Okay, let's continue. In carbon fixation, the enzyme that catalyzes the reaction is rubisco. Carbon dioxide reacts with which molecule to form a 6-carbon molecule, but it's not shown in the picture. But this 6-carbon molecule will immediately be broken down to 3-carbon molecules. So it describes this step right here. So the carbon will react with the ribulose biphosphate, so RUBP. And then the 3-carbon molecule is actually over here. And you can see 1, 2, 3, right? 3 carbons, so that's PGA. So again, you don't have to memorize the names for each chemical because everything else is on this picture. So you can just give me the abbreviations. In stage 2, the reduction, PGA is reduced to what? Reduced to what? By what? So this is PGA, PGA over here. So you can see it reduced to another 3-carbon molecule called the PGAL. So it's reduced to PGAL, and who provides the electrons? So that's going to be the NADPH. 1 molecule of pGal or G3P is used to make glucose. So the majority is used to regenerate. So 1 molecule is used to make glucose, and then 5 out of the 6 will go back to the cycle and regenerate that ribulose biphosphate, right? So you have the raw material to do the next cycle. So this is going to be ribulose biphosphate. How many carbon dioxide molecules in total are required to make 1 glucose molecule? So we'll talk about that. You need 6. All right. So this is an overview picture on the process of photosynthesis. So again, this picture has 2 processes, 2 steps. Step 1, light dependent reaction, and then second is light independent Calvin cycle. So in the light dependent reactions, what you're trying to make are 2 things: the molecule that will provide energy for Calvin cycle, and then you also need to make the electron carriers that will reduce carbon in the Calvin cycle. All right, and then when you start the Calvin cycle, the carbon dioxide just comes from the air. Okay? That's it. That's all the 3 things you need to make a glucose, and then that's a cycle, right? So you start from here, you make sugar, and then some of the raw materials will go back, okay? And then you can do this cycle as many times as you can. All right, so that's the photosynthesis process.